Hematology quiz review in no particular order. Pappenheimer bodies are red blood cell inclusions which are seen in sideroblastic anemia and contain high amounts of iron. MCV is found by multiplying hematocrit times 10 and dividing by red blood cell count. MCHC is found by multiplying hemoglobin by 100 and dividing by hematocrit. MCH is found by multiplying hemoglobin by 10 and dividing by red blood cell count. Basophilic stippling is composed of RNA. Howell-Jolly bodies are composed of DNA. Cabot rings are composed of remnants of mitotic spindle. Pappenheimer bodies are composed of iron. Heinz bodies are composed of denatured hemoglobin. Prussian blue stain will stain iron. Schistocytes are present in microangiopathic hemolytic anemia, prosthetic heart valve replacement, and thrombotic thrombocytopenia purpura. Bart's hemoglobin is associated with alpha thalassemia major. Basophilic stippling would be seen on a blood smear for a patient with lead poisoning. Thalassemias are characterized by a decreased rate of globin synthesis. Alpha thalassemia major findings would be numerous nucleated red blood cells, marked anisocytosis, hypochromia, variable polychromasia, and macrocytes. The findings in beta thalassemia major would be marked anisocytosis, poikilocytosis, numerous nucleated red blood cells, microcytes, hypochromia, dacrocytes, codocytes, basophilic stippling, schistocytes, and polychromasia. Hemoglobin C disease findings would be codocytes, rod-shaped crystals, and basophilic stippling. Alpha thalassemia is most frequent in Asian or African descent populations. Macrocytic normochromic red blood cells, acanthocytes, and stomatocytes are associated with severe liver disease. Beta thalassemia major is most commonly seen in Mediterranean populations. When the oxygen dissociation curve is shifted to the left, oxygen is not readily released into the tissues. After the removal of red blood cells from the circulation, hemoglobin is broken down into iron, porphyrin, and amino acids. The laboratory findings consistent with hemolytic anemia are increased serum lactate dehydrogenase, LDH, increased catabolism of heme. Hemolytic anemia is associated with the breakdown of heme and lysis of red blood cells which have high levels of lactate dehydrogenase. The abnormal hemoglobin that gives green blood is sulfhemoglobin. Carboxyhemoglobin levels are checked in carbon monoxide poisoning. Iron 3+ is bound to hemoglobin in methemoglobin. Transferrin transports iron. On a smear, evidence of hemolysis would show as nucleated red blood cells and polychromasia. The globin chains that are present in Gower-2 embryonic hemoglobin are 2-alpha and 2-epsilon. In order for hemoglobin to combine reversibly with oxygen, the iron must be in the ferrous state. Erythrocyte cell production is stimulated by erythropoietin, EPO. Bone marrow's cellularity is expressed as a ratio of bone marrow cells to fat. Embryonic hemoglobin types are Gower-1, Gower-2, and Portland. After the bone marrow takes over production of blood cells, stem cells remain active in the bone marrow. Interleukin 5 is most responsible for eosinophil differentiation and release from the bone marrow. At the age of 18, the red to yellow bone marrow ratio is 50-50. Fat is yellow marrow. Bone marrow consists of fat, blood vessels, and nerves. Erythrocytes are the cells for the transport of O2 and CO2. In the normal adult, the spleen acts as a site for removal of imperfect and aging cells. The lifespan of a red blood cell is 120 days. Plasma cells are B lymphocytes. When a segmented neutrophil has 8 or more lobes, it is called hypersegmented. Basals have purple, dark blue color granules in their cytoplasm. The organs of hematopoiesis in fetal life are the spleen and liver. Eosinophils have an abundance of specific or secondary granules. 45% of whole blood is erythrocytes. The order of maturity of neutrophilic cell lines from immature to mature is myeloblast, then promyelocyte, then myelocyte, then metamyelocyte, then band, and then segmented neutrophil. The megakaryocyte gives rise to platelets. The order of maturity of erythrocytes from immature to mature is rubroblast, then prorubrocyte, then rubrocyte, then metarubrocyte, then reticulocyte, then erythrocyte. Upon entering the tissue, the monocyte becomes a macrophage. An anemia would be classified as pernicious if there is a B12 or folate deficiency. MCV is the measure of size for the red blood cell. MCHC is the measure of color of chromasia. A decreased reticulocyte count could represent ineffective erythropoiesis. In megaloblastic anemia, the red cells are fragile and have a shortened lifespan, and are prematurely destroyed in the bone marrow and the peripheral blood, leading to anemia. The MMA test is used to evaluate vitamin B12 malabsorption. Methemoglobin contains a ferric oxidized form of iron, iron 3+ to be exact. The basophil has large, abundant violet blue or purple black granules. The plasma portion of whole blood is 7% protein. The lymphocyte has a round nucleus with dense chromatin and blue cytoplasm. The major erythrocyte production site is the bone marrow. The normal sequence of blood development is the yolk sac, the liver and spleen, and then the red bone marrow. The monocyte has a convoluted nucleus with lacy chromatin and grayish ground glass appearing cytoplasm. Yellow bone marrow will decrease if cell production increases. When hemoglobin binds to oxygen, it makes oxyhemoglobin. Deoxyhemoglobin is hemoglobin with iron in the reduced state but not bound to oxygen. Carboxyhemoglobin is hemoglobin combined with carbon monoxide. Carbaminohemoglobin is hemoglobin combined with carbon dioxide. Methemoglobin is hemoglobin in which iron is in the oxidized state. Metamyelocytes have indented nuclei. Myelocytes have round nuclei and may have a flattening side. Increased polychromasia is evidence of a hemolytic event and is present in the peripheral blood smear. The eosinophil has large orange granules. Hypersegmented neutrophils have 6 to 7 lobed nuclei. The only cell normally made by extramedullary hematopoiesis are lymphocytes. In the mesoblastic phase, primitive cells form blood islands that carry on fetal erythropoiesis. The hepatic phase is a production of erythropoiesis in liver, spleen, thymus, and lymph nodes, with production of other blood cells appearing later in this phase. In the myeloid phase, the production of most of the blood cells takes place in the bone marrow. The characteristic morphologic feature in lead poisoning is basophilic stippling. Macrophages are monocytes found in the tissue.